Hello, good afternoon, everyone. Thank you all for coming to the Waterloo Institute for Nanotechnology's Distinguished Lecture Series. As you know, that these series will count for students who need signatures as part of their degree requirements. And of course, our reception will be held after the talk and questions in the immediate foyer in front of the windows before you hit the doors. So thank you all for coming. And actually, our students, please come this way. Oh, no, I guess they want to stay upstairs. It is my pleasure to have our guest, Professor Luke Lee, and I would like to invite Professor Carolyn Wren from the Department of Mechanics and Mechatronics Engineering from the University of Waterloo, who is also one of our distinguished WIN members, to give the introduction and biography. Welcome.
Yeah, thanks everyone for coming to this distinguished seminar series. It's our great honor to have Professor Luke Lee from Berkeley to give us this very exciting talk. So I'll give a brief introduction about Professor Luke Lee. The CV is too long and the biography is very long. I give a very brief one. So yeah, Professor Luke Lee and is a distinguished professor at the University of Berkeley, and he did his degrees down there. He also was a chair professor in systems nanobiology at the ETH Zurich from 2006 and 2007. He was also founding director of the Biomedical Institute for the Global Healthcare Research and Technology in Singapore. So he's a fellow of the Royal Society of Chemistry, and he also a fellow of American Institute of Medical and Biological Engineering. He has published many papers and more than 60 patents, and he's also received millions of the awards. Here, just mention a few of them. It's the IEEE William Morlock Award, NSF Career Award, Fulbright Scholar Award, and a Ho-Am Prize, which is very prestigious. People say it's kind of equivalent to Nobel Prize. So let's give the floor to Professor Lee. Thank you.
Okay, thank you for kind introduction. And this is my first time in Waterloo, and I was very impressed by your university and philosophy. Especially, I'm very fascinated by your training undergraduate student with co-op. Even though I study biophysics at Berkeley, I happen to work in industry more than 10 years and then become engineer. So I was a scientist and to be brainwashed to look down engineer, but now I look up engineer because industry really give me a different perspective. So you will see sometimes I touch the fundamental if I need it, but I have a clear motivation for each topic. So today I was trying to talk about only two topics. Ultra-fast PCR-based SAND. And SAND is nothing but a speedy analytical nano-optofluidic diagnostic system. And then organoid MAP. We want to map the organoid. MAP is nothing but a microfluidic analytic platform. But then I realized that I might not able to come quite often. So I'm going to show you some of the diverse project briefly. And you can raise your hand and stop me if I go too fast. So some places I go slow, some places I go fast. So I like to just get an idea. How many chemists we have here? Few? Okay. How about physicists? Okay, there are a few. How about electrical engineer? Great. Mechanical engineering? Wow, balance. Okay. Bioengineer? All right, thank you. So I'm trying to get an idea which one to go fast, which one go too slow, but I realized that it would be very interesting to handle this situation, but let me try my best. So I have to really thank for my students and postdocs, as well as visiting professors from many different countries. And I like to really thanks for good undergraduate student. I intentionally trained them in during their sophomore, junior, and senior, so that they can prepare to go to industry or MD PhD program or PhD program. And then I really thank for all different research funding agents, including NSF, NIH, as well as the industry. So our group is known as a BioPOS group, bio-inspired photonics, optofluidics, electronics, technology, and science. And it is due to the fact that even though I study biophysics at Berkeley, I end up working in industry making 35 compound laser. So I picked up photonics from industry. Optofluidics we developed is while I was an assistant professor. And electronics, we cannot ignore electronics because all the sensing part, we really need electronics, whether you like it or not, to interface with the biological system. And I'm glad that we can make a new technology in order to develop new science. So this is our group. And motivation is to really transform and shape the future healthcare by creative convergence of science and engineering, medicine, as well as art for humanity. So we want to really work together with many different collaborators, not only engineers, but also chemists or physicists who like to do fundamental study for clear objective. And we want to solve the problem because the medical problem is an ill-defined problem. It's not a well-defined problem that you can find in the textbook in the example. So we have to think about how to address ill-defined problem for developed and developing country. So we are making patient-oriented engineering medicine on a chip. So we want to show you this is not this particular design is basically a technical poem that you want to print in the chip, but I'd like to share with you real poem that really summarizes my research in 4 lines. How many people remember William Blake in high school? Anybody? No? I thought Canadian really read a British poem. William Blake predict late 1700 that we will do nanotechnology. First line actually reflect his prediction. He's very prophetic. He asked us to see a world in the grain of sand. When I read this, when I was in high school, I didn't get it. But somehow, 10 years ago, somehow it dawned on me that, oh my God, he really predicted we can make a nanoscale sand or a micro sand to see a world. So that's what I'm going to share with you. And if other 4 lines, I will try to explain how I interpret this, not only to see a world in the grain of sand and heaven in a wildflower, and whole infinity in the palm of your hand and eternity in an hour. So in this whole infinity in the palm of your hand, I want to educate undergraduate student or graduate student to really use your hand. Instead of using only your computer, the game, you can use your hand to really invent many new innovative science and technology. So it is in your hand, so you can think about infinite number of invention as well as a new discovery. So first, let's just go one by one to see a world in the grain of sand, whether it's possible or not.
To see the world in a grain of sand. Is it possible to gaze at the health status of humanity and the earth in a grain of sand? Can we create smart sands to view DNA and RNA fingerprints from our blood and the earth in real time? Reflecting on Blake's poem in our fast-paced world provides an inspiration to find a proactive solution for our global biosecurity. The world is more dangerous today due to the emergence and spread of new infectious diseases. With smart sands, we can prevent such outbreaks from happening through early detection. We propose SANS, speedy, analytical, nano-optofluidic diagnostic systems. Smart SANS are on-chip for rapid and accurate molecular diagnostics. Smart sands can be used by anyone with a smartphone, even in areas where rapid and accurate diagnostics are usually inaccessible. Smart sands will be globally connected to an integrated data hub. Smart sands rapid and accurate molecular diagnostic network for human, agricultural and environmental health will radically improve global healthcare and empower us to create a new proactive, predictive and preventative paradigm for enhancing global biosecurity. Is it possible to gaze at the health status of humanity and the Earth in a grain of sand?
So that's one of the reasons that I went to Singapore to establish the new institute to really execute this mission. But there are many issues, politics, so I failed. I submitted this proposal 7 times, they reject 7 times. So I came back to Berkeley, but I didn't finish my mission. So whether you can help me from your part, from UW student or whatever means, we'll execute, but I'll share with you whatever we did to do this. So last 17 years, start with making a miniaturized confocal microscope. This is our first smallest microscope. But as you know, Silicon Valley is well known for, semiconductor industry. So nothing but sand. They use a sand to create microelectronics. Now it's a CMOS camera is getting smaller, and it's time to think about how to integrate this kind of technology for healthcare diagnostics. So let me just introduce a few examples of the bio-inspired photonics for smart sand. Let me start with like millimeter scale. So in this area, we develop some device, but let me just introduce only one of them that we learn from the nature. This compound eyes from insect is fascinating. There's 2 eyes, but in the big 2 eyes, there are many, many small eyes. It's the compound eyes allowed to take all the omnidirectional the image. So this image collection from insect we can learn, but how do you really fabricate this kind of compound eyes is challenging, but as long as we learn this, there is the lens and crystal cone and like waveguide structure like this rhabdomer. So there's kind of fascinating biology. It starts with the cell and basically information from gene. How do they do this? So we didn't start with the cell, but however, we use polymer to create microlens and then focus down with a photopolymer to form waveguide structure and make it similar to natural ommatidium, so that we can create thousands of compound eyes on one big lens, so they can take advantage of this omnidirectional imaging and then capture all this biological information. But here, front part is a lens and back side is waveguide structure. And then you can take advantage of this imaging system. And then you can build a nice remote system, detection system with the CMOS camera. And this is artificial compound eyes. So you can spread this in many different places. For me, artificial intelligence is kind of fashion, but we want to get real data, not artificial data. So it is really artificial or real intelligence. It's up to you, but everybody's following artificial intelligence, which was already existed 30 years ago, but we were revisiting. Nothing wrong with revisiting, but we can learn from the nature. This is a diatomic structure. Seashore you can find, and they form this from cellular structure. And then we can take advantage of this kind of structure and learn from it, and then generate a lot of interesting, smart sensing modality for environment as well as a body level detection. Now I'm going to show you nanoscale structure. Can you see something? Can you imagine? Is it from galaxy? What do you think? Looks like, right? When you do research, you have to really get out of your typical bias. But this is not a galaxy, but this is nothing but plasmonic inside of cell. So it's question is it possible to see a cellular galaxy with a grain of sand? This is a really tiny, tiny grain of the dust, okay? Nanoscale dust. But we want to use this kind of dust to see real electron transfer inside of the cell. But we need to really we need to ask whether there is any real electron transfer or not. You probably studied this photosynthesis system in high school, that the sunlight and generate photon, and then photon excite this photosystem 2, and then it pumped, and then it passed down to electron to photosystem 1, and then you generate basic biochemical reaction. If you're a physicist, can you believe that there's a real electron transfer in cell membrane? If you're a physicist. If you're a chemist, how do you interpret this as whether this kind of the textbook information is real or artificial? You probably study and memorize and regurgitate and pass the exam. Did anybody measure this? Where is the evidence that somebody poked with a wire and measure electron transfer? Did you ever question this? I didn't question either before. But one day I started questioning why there are so many biology has hypotheses, but never proven. Like DNA become RNA, RNA become protein is a central dogma of molecular cell biology. Is it true? Is it true? People believe that it's true. But the person who published this didn't do any experiment. He's a very smart physicist and full biologist because he got Nobel Prize. That doesn't mean that he completely understands biology. He said protein never influenced DNA, but you know that transcription factor influenced DNA. RNA can influence DNA, RNA can influence RNA. So partial truth is not 100% truth. So even here, it's very logical that there's electron transfer, but where is evidence somebody measured this? I couldn't find any paper. But I found DNA become RNA, RNA become protein as a central dogma. Dogma is actually faith. It's not a scientific word. But he wanted to create that this is the faith that you have to believe. And he published in Nature. So everybody quote, and it's in the textbook. Yes, there is partial truth, but it's not 100% truth. Because the protein can influence, transcription factor can influence DNA. So same thing for the even electron transfer. It is very logical, but is it really quantum biological electron transfer is here? So it's a question, then we have to solve this issue. But many people logically explain. Even I believe that logic, and I believe that, but I didn't see any evidence that there is a quantum mechanical electron transfer in the photosystem or DNA repair and respiration and so on. It's very logical and published in Nature. So we have to think about how do you capture then emotion. When I say emotion, it's electron motion or electron transfer. So we have to think about, this is beautiful fluorescent image of the cell, but we have to think about how to capture real emotion, not the makeup image. Yes, we need to have a makeup image to distinguish all different compartments of the cell, but we have to think about how do you capture cellular signaling pathway in real time. So we have to think about making new way of exploring the cell using small, tiny satellite. So I'd like to just share with you this interesting history because I happen to work in the company called TRW. It's a defense electronic company, make all this satellite. So when we receive this beautiful image, now you see this as a what? Galaxy, right? But do you know our reaction? What was our reaction when we received this beautiful galaxy using satellite? At the time, we were tension between USSR. So when we have a psychological tension and stress, we interpret this, each dot, as a nuclear plant from USSR. And you don't know because people hide this, because there was an overall reaction for a certain time, and then they realized that, oh, that's not true, but it took some time to interpret this as the galaxy. So what I'm saying is when you do science, you have to really get out of your typical bias mode. So we can think about how to put this nothing but plasmonic antenna inside of the cell, and then think about how to capture electron transfer in the vicinity of this tiny antenna. So this plasmonic particle with a different shape means a different resonant frequency. But let me just briefly tell you, for someone who didn't study very simple surface plasma polariton, if you excite with the light, in between this dielectric layer or in a metal layer, you can generate a localized coherent charge oscillation due to the coupling of the light and then the surface charge, so that you can generate different wavelengths due to the permittivity of this metal, different metal, gold, silver, and aluminum, and platinum, and so on, and as well as the different permittivity of the dielectric layer, either DNA or protein or medium. So you can take advantage of this surface of plasma polariton. And then another advantage is that you can think about focusing, even, let's say, wavelengths of 1 micron, you can focus down to even 10 nanometer. Is it possible to do this with the conventional optics? You cannot focus typically lambda, less than lambda of 2, but you can basically kind of cheat it with image processing to make it narrow. But physically, you can think about a different way to focus this light. But it's very fascinating that even the artists even think about connecting in wave and plasmonic shape. I hope you remember this picture. Why I'm bringing up this art, because I want you to enjoy why you are in the technical school or science major. Enjoy and see why this particular artist linked with this wave and coupled with the crescent shape. It's kind of fascinating to reflect that whether artists or physicists, people want to link. So we want to link light specific wavelengths, and then if there is a DNA attached or RNA or peptide, you can basically excite on the surface electron and then interact with a neighboring biological molecule. So let me show you as a practical example. We can attach DNA or RNA or peptide and deliver by endocytosis. And different shape of the crescent with a different angle means a different resonant frequency. So let's assume that you attach specific sRNA onto this antenna, which has a 980 nanometer the resonant wavelengths. Then you can deliver this specific sRNA, and then if you want to liberate them to do gene regulation, you can use a phone outside. Basically, you use a specific wavelengths to light, then you can resonate and then liberate this without damaging the cell, because you don't need to pump a lot of light intensity, because you can resonate only the surface, right? The specific antenna. So you can take advantage of liberating attached the DNA, RNA, peptide, as well as the sensing. Okay, so that's the reason. But in terms of the real electron tunneling, it's a question whether can we really measure any electron tunneling or electron transfer. This is the new way of checking the electron transfer of the enzyme in living cell. So quantum biological electron tunneling is basically a simple way to capture oxidation reduction in living cell, but let me just briefly mention, quantum biology is nothing but just quantum mechanical study of the electron in biological molecule. So as long as you study electrons in biological molecule, and then think about tunneling. Is it really possible to transfer electron from a neighboring protein is a question. So there are many people did theoretical work and many people discuss, but it's time to visit what is the simplest way to study. So this is antenna, nothing but plasmonic particle. And then you couple with, in this case, enzyme, which is very important, cytochrome C, which play very critical, the enzyme play. Then you can link with a specific linker molecule and then study whether electron, hot electron that you generate from this antenna can transfer. Then how do you detect whether there is a really electron transfer or not? By looking at this quantized spectrum, you can identify whether there is an absorption peak is recognized or not. So that's the one way. We didn't poke with the wire, but we are looking at the specific resonant peak. So basically, I'm explain again. If there's a protein in the vicinity of this plasmonic antenna, let's assume that this protein has this absorption peak. Without this protein, if there's no right absorption peak, you generate this kind of scattering light. But however, if there is a protein which has this kind of absorption peak, because of the energy transfer, resonant energy transfer, or electron transfer, you can see quantized resonance quenching dip can be observed. This specific wavelength is matching with this electronic state of the absorption peak. Otherwise, if there's no electron transfer, it has a broad peak, and you cannot recognize this quantized resonant quenching dip. So to verify, we test with the different lengths of this linker molecule and then see whether we can observe basically electron transfer can be observed with the different lengths of this carbon chain. This is for the reduction peak of the enzyme, and this is the oxidation peak. You can see as you decrease the distance between this junction between the antenna and enzyme, you see increase the signal of the electron transfer of the reduction of cytochrome C, and this is oxidation cytochrome C, and so on. So then what do you do with this? We deliver this inside of the cell and then try to understand whether we can capture electron transfer in real life. So by monitoring this critical protein, the cytochrome C, during this electron transfer in the mitochondria. Mitochondria is an energy source, as you know. So we test in vitro first before we put the cell. By comparing with this bulk solution, you can get nice peak from oxidation, reduction to oxidation can be observed. And the same thing can be done in a single particle base from single peak, which is oxidation, and the double peak is reduction. So after you accomplish this in vitro, and then you put this inside of the cell, and during the cell death, apoptosis, how this double peak, which is a reduction peak, can increase, and single peak is increased in apoptosis, but in necrosis. Very fascinating to observe that these two peaks, the red and blue peak, is reduction, but it oscillates while the oxidation peak is increasing. So this is the beginning of this electron transfer imaging in living cells. So hopefully we'll continue to demonstrate how we can capture electron transfer of the enzyme in living cell condition. Okay, next line, and heaven in the wildflower. There are many things to see in the wildflower, but of course, when you look at this, it's like a nice heaven, but I see nice symmetry of the leaf, the petal, but I see more interesting symmetry in plant virus. Inside of the plant virus, we have this nice, beautiful symmetry. So we can think about this as a bio-inspired optical antenna. why I look at this as an antenna, what's the advantage of this is following. We can harvest this plant virus from plant, and then you decorate with the gold and then generate a half side as the antenna, and then put in vitro diagnostic, molecular diagnostic, or in vivo imaging, so that we can take advantage of this so-called hotspot generation of this through this topology for surface-enhanced Raman scattering, or PRET that we mentioned, that plasmon resonant energy transfer. So optically, you can simulate and then think about which particular topology is better to generate so-called hotspot. So high electron density generate more light, right? Information, scattering light. So we simulate with the different topology of a plant virus, and then we can characterize optically and see which particular geometry topology is ideal to operate in 1 micron the wavelengths to excite. So the tomato bush shunt virus or cow pea mosaic virus has a similar peak, and then you can take advantage of this cow pea mosaic virus or tomato bush shunt virus to use it as a sensor. So you can take advantage of surface-enhanced Raman scattering compared to a smooth surface. This topology allowed to improve from micromole to femtomole sensitivity, as well as in PRET, you can improve the sensitivity due to the this kind of topology. This looks smooth, but we're dealing with spikes of this peak in nanometer. But imagine, can you really fabricate with your electron beam or the 3D-dimensionally this kind of virus structure? It's still questionable, but in nature already generate a beautiful topology like this, so you can take advantage of existing natural plant virus as a sensor. Okay, whole infinite in the palm of your hand. As I mentioned from the beginning, it's your hand. How many graduate students are here? Great. About undergraduate students? Oh, fantastic. I'm really glad to see many undergraduate students in this audience. I really resonate with the philosophy of the University of Waterloo, the training undergraduate student. So it is your hand. It's up to you whether you want to make more new invention or whether you want to play game only, right? So with that in a context, let me briefly go over history of computing machine, because I want you to think about a biological computing machine or a biological machine that allowed to make a quantitative biology. But we need to go back to a history of abacus. You might wonder, how does abacus relate with the microfluidic? But it's very fascinating to see history from Chinese abacus or Roman. They can argue which one is first, but I want to just highlight that Pascal's calculator, as well as ammoniac, ENIAC, really make huge progress. It took a long time. But when I went to America, 1976, this DEC computer was the best computer. While this Steve Wozniak computer looks ugly, but this best computer, if I had a time sharing 1 hour, I was a lucky guy. But why this digital electronic corporation disappear? Do you know what the reason? PC. PC? Good, excellent answer. The founder, Ken Olsen, was as famous as Steve Jobs or any rich entrepreneur. He was in Fortune magazine, but he said there is no reason for any individual to have a computer in his home. He didn't have a vision for a PC, even though he could really reduce, but he didn't want it. So he didn't have a vision. So it's very important. So if someone is challenging you, whether you're undergraduate or a graduate student, and if you're working on some projects that why you are doing this, and then somebody ridicule you, just hang in there, because there are many incidents like this This is a history of the transistor. One transistor from 1952 to billions of transistors. It took some time. But not only that, the person, he's most famous, one of the most famous physicists in 1931, Wolfgang Pauli said, one shouldn't work on semiconductors. That is a filthy mess. Who knows whether any semiconductors even exist? He didn't even believe, right? He's a very famous physicist. But he didn't even believe. But some engineer made a progress. Ironically, Faraday even studied semiconductor effect. It took a long time, but there's an important role industry play. Jack Kirby, he didn't have a PhD, but he got Nobel Prize in physics in, I think, 1990, I forgot year. But the other two also contributed. Same from industry. But why only Jack Kirby got a Nobel Prize, even though he got only bachelor degree without PhD? These two gentlemen got a PhD, but they didn't get the Nobel Prize. Can you guess why only Jack Kirby got Nobel Prize in physics? Who didn't get? He was an electrical engineer, but he got the Nobel Prize in physics. It's a very simple reason. He lived longer than these two gentlemen. So you have to take care of your body and then wait for the right moment, okay? So this is very interesting to compare solid-state silicon IC versus soft-state silicon with the E. It's nothing but PDMS or just elastomer. The equation is completely different. A lot of people tease that, oh, microfluidic is just fashion. But somehow, even though I study physics and industry at that satellite company, I worked on actually SQUID, superconducting quantum interference device. That's why I'm really familiar with the tunneling junction. But I didn't want to do this SQUID that operate at low temperature. But this microfluidic require extra work. It's really messy. But again, the soft silicon IC, which is microfluidic, is moving along really fast because of the semiconductor, the process that we are adapting. So I'm going to just introduce a few examples. There are many wonderful examples, but due to the time, I'm going to highlight a few examples from our side, from my lab. First, this is optofluidic, but then the patch clamp array or dynamic cell culture and single cell analysis and liver on a chip early 2000. So I'll show you some highlight. So we call it as an integrated microphysiological analytic platform. So we developed first dynamic cell culture and live on a chip, and then Harvard made it long on a chip and so on. So this moving really fast of organ on a chip. And the idea is how do you really see the body function by using your own cell and then reprogram this IPS, induced pluripotent stem cell, and then culture them, and then analyze what is the best drug for individual. So that's the motivation. So let me just go over some history, the physiologically relevant dynamic cell culture system. So why I decide to do this? Because when I see this is an invention by Petri, this is fascinating. History is fascinating. Because of Petri, he's supposed to get Nobel Prize. But Petri got more recognition, right? He didn't get Nobel Prize, but everybody knows Petri did. How many people know Professor Koch got Nobel Prize in bacteriology? Some people, yeah. But this is a really good combination. So if you're an engineer, you work with a physicist or chemist or a biologist, help them. Even though you think that you are a servant for them, but play the right role and then get recognition. Petri this is an invention. But then somehow I realized that, okay, this is a good invention at the time. But this is saying that Earth is flat. Our body is not static. this cell culture, Petri dish 196 was still static. Some bacteriology and microbiology, that's okay. But you have to think about this static culture without perfusion and washing this. For example, in our brain, if you believe that Alzheimer's disease is caused by A-beta plaque formation, you have to think about how A-beta can be cleared out. You need to have a flow. If there's no flow, you cannot clear out this junk. Whether it's an interstitial flow or circulatory flow, there's flow. But here, there's no flow. And then you accumulate this. Of course, you can use pipette to wash away, but then you have to think about how do you make a cell and then make a perfusion channel. So basically, if it is tissue like this is a tissue sample, and there's a circulatory flow. In between circulatory flow, there's an interstitial flow. So you have to provide a similar design. I'm not saying this is the best design. You can make a better design. So there's small gaps so that you can have an interstitial flow while cell is loading here, while you can have also circulatory flow. So you can control all different conditions of the flow. And then you can lay out the design with the computer and CAD. And then you can inject different concentration gradient and different flow and so on. So you can have a multiple experiment simultaneously. But why I'm just showing you this? Because there are many temptations. So even a few months ago, Shinya Yamanaka, who got Nobel Prize for the stem cell, has to apologize because one of his postdoc fabricated data and had to retract the Nature paper recently. Okay, and then a couple years ago, another developmental biologist suicide because his postdoc fabricated data, Science and Nature paper, so he couldn't handle. He was one of the most prominent developmental biologists in Japan, in the world. But he suicide because it was, shame that his, postdoc fabricated data. And same thing happened in Korea, but it's happening. I'm not defending for them. They probably see once in a while. But why they couldn't reproduce? If you think that the Earth is still static, you have to think about this. You have to provide them nice control of shear stress, nutrient content, and there are many things to modulate, but you have to really control with microfluidic device or macrofluidic. Doesn't have to be microfluidic as long as you provide similar way to recapitulate the body. So let me show you one example with the brain. In the brain, if you culture the brain cell without this kind of network, you have to question. So current mini brain is like this. You publish well, and then you use all this conventional method. They use a bioreactor so you can rotate, but then it's a different stage. So if you do drug screening on this, then how do you know which stage is the right stage to perturbate with the drug, right? So you have to think about real stability and reproducibility of this mini brain. So we decided to make a better way to make a reproducible structure using this kind of dynamic interstitial flow. So this design has a hemispherical shape and perfusion channel, and we have also electrode to make non-invasive real-time monitoring of pathogenesis dynamics. And this is a top view and cross-sectional view, but there's a main channel hydrodynamic allowed to form the mini brain structure, and this extra channel perfuse the perturbations. And basically, we can form very similar shape, right, due to the hydrodynamic and this design. And then we can also study case with the normal neuron and astrocyte. In this case, we are mixing with the normal neuron and normal astrocyte versus to normal neuron and inflamed astrocyte. What if like mother has inflammation or infection? How does it connect with the neural network? You can see the brain size even changed due to the simple inflammation, just one factor. Due to the time, I'm going to skip a lot of detailed proteomics, but I'd like to highlight this electrophysiology. You can monitor from day 1, 2, 3, 10, and get huge data. It's a really, to me, it's not artificial data. It is real data, but it's a lot of data, but we want to analyze this. This is normal, and this is a case with LPS treated. Why this case is different? So you can also induce with the PZT and then see this epileptic behavior. So you can take advantage of this kind of system to study even basic organogenesis, and then think about some possible Chinese herb or any natural herb to perturbate, so that when you induce perturbate, and whether this abnormal behavior can be normal behavior, you can observe while you are watching this. So since we have a perfusion channel, we can pulsate this and then monitor detail of this electrophysiological activity. I'm going to skip pancreatic. So you can form pancreatic, and then you can do a lot of detailed study. But I like to just highlight kidney, because we use IPS. Using IPS, we can receive the cell at 9 days, and then we can control actually different pathway. Again, because of this perfusion channel, we can induce intentionally more to stimulate to generate more glomeruli or tubular structure by controlling the pathway. So you can see more tubular emphasis organoid can be formed, and compared to this normal spherical shape, you can get asymmetric geometry, and then you can make a polarized compartment here, and you can really study detail of like cortex. So you can see you have a more glomeruli in one side, and then you have a proximal tubule and distal tubule and so on. So this kind of control, modular activity can be done in microfluidic platform. So you can take advantage of this basic study of organogenesis, as well as drug screening in this environment. And you can see actually tube formation inside of this here, so that you can take advantage of this kind of system. I'm going to skip this. And in terms of also disease, so you would take a patient cell, and this patient has this autosomal recessive polycystic kidney disease, ALPKD. Then using this patient cell, and then you can study whether we can observe the cyst formation. And this is normal case, and this is the patient case. And then you can think about then, okay, it's good to see the cyst formation and observe this spontaneous cyst formation. But at the same time, you can challenge with a different drug and see whether you can remove this, right? So actually, since it's not published, I didn't put it here, but there's a very fascinating result. By controlling signaling pathway, you can actually remove this cystic formation and so on. So remaining 5 minutes, I'll talk about diagnostic. So I happen to talk organoid first, but molecular diagnostic is something that I want to disseminate this chip. So our idea is a sample to answer device at low cost. So we want to really use blood, and then we can separate. So you drop the blood, and you can separate plasma. And downstream, you can do PCR, or you can do the protein ELISA analysis. But as you know, this is what we like to have, like affordable and sensitive and specific and user-friendly and robust and equipment-free and deliverable. So our simple device is a most simple one. There's no external pump. It's a self-powered fluidic device. Let me show you that. This device, we integrate everything. So including the amplification initiator that is patterned on top of this fluidic chamber. While we are separating plasma, there's an amplification initiator, and plasma is reacting so that you can initiate isotomal amplification. So each red dot is a reaction chamber, so you can observe nice, simple way to make a one-step digitization. As you can see, there's no external pump. We built a vacuum battery. It's not a capillary-based because it's all hydrophobic PDMS. But you can see immediately you separate individual chamber. At each chamber, we are doing the DNA amplification. Okay, so this is a degas-based vacuum battery, so that we don't have to use any external pump. And then this animation shows how plasma is pumped this in degassing chamber, so that you can avoid any bubble trap. And this is a failure case without proper design. Everything is connected. But if you make a micro clip with a 40 micron height, then you can isolate individual chamber. So, and then this is showing that the real blood, this all the cell is going through, and some of the cell can be trapped, but this is plasma that you trap in the individual chamber. And then with this concept, we can amplify. This is MRSA DNA. We use RPA. This is isotomal amplification. At this moment, we can detect 10 copies per microliter. So this is the detection limit. But this is a good demonstration from sample to answer, right? Blood can be separated on chip and the amplification of the DNA. But I'd like to share with you an eternity an hour. I can die in the street any moment. But I remember my daughter, when she was 1 year old, she has an infection, but the doctor didn't know what kind of infection she has, so they'll keep adding antibiotic without knowing any specific signature. So we really think about rapid and precise molecular diagnostic. It's not only infectious disease. Definitely infectious disease is important. But even cancer, you don't have to get rapid, but we have to think about rapid screening. So we have to think about like a very fast and precise molecular diagnostics. So we decide to work on ultra-fast photonic PCR. So we can get inspiration from the nature. Of course, here I was questioning also photon to energy transfer to electron transfer, but we can think about this kind of model in the plasmonic system, and then simply we can oscillate electron in the surface to heat it up and make a very fast heat engine to speed up this gene amplification. So compared to conventional PCR chamber, which use a big heater block, it uses huge power and it takes time. But here, simple plasma, and then we use LED instead of laser because you don't need to use a high power intensity because you need to resonate with the right the frequency. And then simple LED allow to basically heat it up due to the electron scattering. It's very simple physics. And then you turn it up the light and then make a phonon vibration and make a heat dissipation. And then you can have a many well underneath of this plasmonic surface. And then you can take advantage of making a few minutes for 30 cycles. So we made this simple system, ultra-fast photonic system that allowed to make less than 5 minutes for 30 cycles. And I briefly mentioned that this crescent come back because this crescent structure allowed to make a plasmon resonant energy transfer based, because the previous one is a flat. We didn't really optimize the resonant frequency. But in this case, we design array of these crescents so that you can have a lot of array. And then you can pick, depends on the size of this crescent geometry, you can tune it to resonate in 600 or 800 and 1,000 or so on. It's just like you don't have to yell at to your girlfriend or boyfriend when you have a resonant frequency, right? You match, and when you do low power. And then when you do that, you can speed up to 56 seconds instead of a few minutes. So hopefully, I give you a big picture that you can make a simple PCR system on a chip, and you can interface with the smartphone to detect. And then now it's time to detect healthy big data, not junk data. Using, again, compound eyes, you can have many so that you can have a high resolution detection of the big data for ultra-fast digital PCR. So hopefully, I'll give you a big picture that it's time to see and understand and act. Okay. So what I mean by that is that there are many data we can collect from environment and food and our body and water, all molecular based, because we are what we eat. We can correlate with food contamination as well as infections, our disease. It's time to think about how to solve this, our health condition by correlating and this information. So I'd like to just share with you for students that one picture is worth 1,000 words. Everybody knows this, right? Yes, it is important. But next one is more important. Understanding is worth 1,000 pictures. You have to understand. Still, I don't understand exactly electron transfer. I'm still questioning, okay? And action is worth 1,000 understanding. Even though you understand, if you don't do anything, it's useless, right? So I'll finish with one philosopher's suggestion. Knowing is not enough. We must apply. Willing is not enough. We must do. And in the realm of ideas, everything depends on enthusiasm. And in the real world, all rests on perseverance. Thank you. Okay. You laugh about it because the plant virus, most plant biologists actually dehydrate. They collect in their drawer. So I collect them from biologists. It's a dry, right? So I just deposit matter on top of it. That's it. Very simple, right?
So that is kind of like a physical...
Yeah, physical deposition. But if you are a chemist, I would recommend to coat it with different, chemical deposition, to make nice conformal coating. And
so in that case, then you coat it like a batch?
In,
then it's just like, for example, like chemical deposition, that it just is a batch, like one shot, and then you coat it?
I'm not chemist, so we fail many times. But the one I show you is a physical deposition. So we are trying to actually assemble together. But we didn't make it with the assembly. But the one I show you is just physical deposition on top of a dehydrated one. But we test, even though it was dehydrated, if you actually put it back to the plant, actually their function is very fascinating. The plant virus.
Thank you.
Welcome. Yes?
One of your slides, you mentioned understanding mitochondria.
Mm-hmm.
Any progress in the role of mitochondria in aging?
Oh, fantastic question. I'm getting old. I like to die gracefully. Like studying aging with the cancer is fantastic, but somehow this idea of capturing electron transfer using antenna, whenever I apply grant, everybody reject. So I never get any grants, so it's like moonlight job. So any student who are interested in doing this, doing their moonlight job, so it's very slow. So people don't believe you can detect this electron transfer still. So that's why progress is very slow, but I do believe, because I see this, but whatever reason, reviewer just reject all the time that there's no point of doing this. I said, if you really understand this apoptosis, and electron transfer. Actually, you can think about using it as a molecular pacemaker. Opposite direction. You can modulate from outside. In 1960, the pacemaker was a the person who did pacemaker was a ridicule. That is totally nonsense. Now you think about tiny molecular pacemaker to pump the energy for the mitochondria. I hope I give you some ideas. Yes.
Questions on top.
Yes. Yes, go ahead.
That's you.
Thank you. You mentioned plasmonic with energy transfer.
Yes.
Would the acceptor need to be a plasmonic material as well?
I'm sorry?
Would the acceptor need to be a plasmonic material as well? So that the excitation frequency for the acceptor would be the same as the resonant frequency of the donor?
Oh, you are saying that...
Similar to fluorescent resonant energy transfer, but with plasmonics instead of fluorescence?
Sounds similar, but in FRED, you cannot capture absorption peak. I hope you understand electronic state of absorption peak. If you use a FRED, it's not specific absorption peak.
I thought FRED is all about absorption peak. And
No, I'm talking about every molecule has an electronic state absorption peak, like fingerprint. We are detecting the fingerprint of the molecule. In FRED, you can energy transfer, but you cannot see fingerprint of the molecule. I hope you catch that. You think about it, okay? And send me email if you have a question. Yes.
I might.
Yeah, okay. Yes.
Those brain cells cultured, are they able to do like reasoning or?
Oh, no. I'm sorry, it's misleading if you think that it's mini brain is reasoning. No, this is the simplest way to test different hypotheses. What if astrocyte is inflamed? Everything is normal, only astrocyte has inflammation. How does it, generate pathogenesis of the neural network? Why there's an interesting epileptic behavior is generating? What's the cause? Then we have to tease out one by one. But you cannot do that with the animal, right? Oh, by the way, the organoid chip, I'm not just doing it because I'm doing microfluidic, because pathologist, one of the famous pathologists, told me that I don't believe animal model anymore. He spent 30 years. Because immunology of the animal and human is different, and the ratio of the neuron and astrocyte compared to mouse and human is different. So it's hard to really correlate. So he's not saying that organoid chip is best, but he is encouraging us to think about how to make dynamic cell culture system that allow to study systematically. That's it. But I don't think that anybody can make mini brain with reasoning.
There is this field of neuromorphic computing that try to define computation based on the way the biological brain is.
We can learn from it. I mean, as you can see, this connection through this junction. Even though I show only junction with the plasmonic and cell, if you can study really electron transfer between the protein to protein junction, it's very fascinating. I don't know how to measure at this moment. Even though I try to poke with the electrode, you cannot detect this tunneling. But if you can understand this quantized data, then you can really think about a better way to generate new computer, right? But we have to learn from the nature with the proper tool.
We'll just take one more question, and we still have the reception. Now you can still talk to Professor Lee afterwards. Okay, sure. Yeah.
Yes.
When you're transferring the energy from an element to a protein, are you able to see the structural changes too in the protein?
At this moment, I don't have a tool to observe unless you denature, right? That I can tell whether it's denature or not, because it doesn't work oxidation reduction all the time, right? But however, this conformational change, we don't have a tool. We don't have a tool to look at the same time while they are doing electron transfer. So you should invent some new imaging tool.
But are you like actually seeing the signature of like changes in absorption or the emission spectra?
Exactly. That's what I showed, that reduction, absorption, and oxidation reduction, the peak is showing. Before, there's no such peak is observed. People never able to see oxidation reduction peak in real cell. You think that is a simple, yeah, it was simple, but people didn't see this simple. The simplicity is the ultimate sophistication. So make a simple detection imaging system. That's the best way.
And one more thing regarding the organoids.
Mm.
When you're culturing the organoids, are you culturing them at like different embryonic stages, or are you like taking the sample, like try to grow the adult brain, like a sample of adult brain, and seeing the molecular diagnostics between the diseased brain and between the healthy brain?
Oh, disease case, we receive the cell from patient. Okay. For normal case, whether it's a diseased patient or not, I mean, we reprogram the cell, iPS, and after 9 days, we form this spherical shape. Either through this hydrodynamic focusing, you can form nice shape, and once you form and then you stop flow, and then you perfuse. It's a different flow. You cannot apply a lot of flow rate because you have to provide similar to interstitial flow. It's a very small flow, but there's a flow to clear out a lot of, shit, right? The cell is releasing their own cytokine.
Yeah, I'm sorry, we have to end here, and we'll go down to reception time period, and I can still try to talk to Professor Lee. Let's thank our speaker again, and Professor Lee give this.
Okay, thank you.